Good morning, everyone. Good to see you.
Morning.
Hi.
Hope everybody had a nice weekend. Yes, if you wouldn't mind, please put your name into chat for me or your nickname, so I have a record that you were here with us today. Thank you. All right. Very good. So today in lab, we are scheduled to talk about respiration, which, believe it or not, is the last biochemical test anyway that we're going to be talking about in lab. On Wednesday this week, we're going to be talking about a lab assignment, which is the identification of an unknown microbe. And this is kind of what we've been working up to all semester. You're going to be given some data, and you're going to use that data to make an identification of a microbe. That'll be on Wednesday. We're also going to talk about some of the tools that I'll be giving you to help you identify that unknown microbe, tools that are called dichotomous keys. So we'll talk about all of that. We'll talk about the assignment. We'll talk about how to use these keys on Wednesday this week. I do not anticipate meeting on Friday, but we'll know better on Wednesday whether we need to get together on Friday. All right. Yeah, so please go ahead and put your name into chat for me if you haven't already. Before we get into material today, I just want to pull up a story that's in the news right now, which actually has a direct link to what we've been learning in class. I'm not sure if you've been hearing anything in your news feed, but there's a bit of an environmental emergency right now down in Florida. Has anybody heard anything about this or read anything about this, about Piney Point down near Tampa Bay? So if you're not familiar, a state of emergency was called
The flood?
Say again?
It's overflowing? Or
Yes. Yes. A state of emergency was called over the weekend because of a leak in a retaining pond at an old plant called Piney Point, an old factory that used to manufacture, among other things, fertilizer. In the old days, many, many decades ago, places like this, factories that produced toxic waste in the form of something liquid, they used to just release it out into the environment, believe it or not. They used to just have pipes leading out of the factory that would just release the toxic materials out into the environment. And after the EPA was established, we started cracking down on that. And we started requiring factories to manage their own toxic waste. One of the ways that they have done that is by digging these massive sort of trenches where they'll line these big areas with some sort of impermeable rubber or plastic, and then they simply fill them up with toxic wastewater. And the idea is that over time, the water in those retention ponds will evaporate and all the toxic stuff will be left. Now, I'm always curious what they plan to do with the toxic stuff, but nobody ever talks about that. From time to time, one of these lined retention ponds will leak and it gets into the news. So that's what's happening down it's in Manatee County in Florida, which is near Tampa Bay. Now, here's why it's interesting to us as students of microbiology. I'll show you a picture of these retention ponds. right here. This is what it looks like from the air, or what it looked like a few days ago. So these are massive, massive facilities you can imagine. You can see the little factory over here. They dig these retention ponds, they line them again with some kind of a plastic or a rubber liner, and then they just send all their toxic waste out into the ponds. This is a very common practice even today. So one of these ponds has sprung a leak. And there were two concerns. Number one was an environmental concern, and number two was a concern for just emergency loss of life, because these ponds hold millions of gallons of wastewater. So if, for example, one of these walls were to give way, all of this volume of water would head out. And there happen to be residential neighborhoods in the area. Lots of people live sort of downstream from these reservoirs. So one of the big concerns has been, oh my gosh, if this thing gives way, people are going to flood, their homes are going to flood, people could drown, a catastrophe. So of course, the only thing that they have been able to think to do is to manage the leak, to allow the wastewater to keep leaking at a rate that will drain the pond enough so that it won't collapse. Again, that will help prevent flooding in residential homes. But the wastewater is being sent out of the pond. And it's going into Tampa Bay. So you can imagine that there are a lot of people who live on and around Tampa Bay who work as fishermen, as tourist, sailors, and people who run charters on their boats. They're very, very concerned because millions of gallons of toxic wastewater are heading out into Tampa Bay. Now let's talk about what's in this water, because this is where the microbiology comes in. Piney Point was a fertilizer factory in its day. It made fertilizer in addition to other things. And in the wastewater that gets generated during the production of fertilizer, there is a high level of two molecules, phosphate and nitrate. And what happens when you put phosphate and nitrate at high levels out into a natural body of water is all of the microbes use it as nutrients, and you get an overgrowth of microbes in that body of water. All of the bacteria and the archaea and the fungi and the algae, they take all of that phosphate and that nitrate and they use it as a food source, and they grow and grow and grow. And when that happens, they tend to kill the higher organisms that are living in the water. So the fish, the shellfish, the other marine mammals. It's a bad situation when you have an overgrowth of one type of organism instead of everything staying in balance. So only time is going to tell how this is going to affect Tampa Bay. But you can imagine all of the people who make their living in Tampa Bay are very upset right now, because that's exactly where this water is headed. It's being released. I'll show you a picture. It's being released out of the wastewater pond. Let me see if I can toggle this. No, not quite. It's being released out of the wastewater pond and allowed to go downstream into Tampa Bay. So this is just an image right here of where the leak is. You can see this wall, this levee is where the leak is. You can see that hole that has developed. And all of that water is streaming out of this gigantic pond. You can see the black liner right here. Millions and millions of gallons of water are being released. A lot of the water is already gone this morning, and it's gone into the environment. So again, it saved a catastrophic flood, because had they allowed that thing to just sit, an entire wall may have given way and people would have been flooded out. They might have been killed. Their homes would have been destroyed. But by preventing that disaster, another disaster has now been created, in that all of that phosphorus and all of that nitrate is going into Tampa Bay. It really makes you question whether or not we should still allow wastewater, toxic wastewater, to be stored like this, or if it's a bad idea. There should be another way that factories manage their waste, instead of just leaving it in these giant ponds where accidents sometimes happen. So yeah, that's going on right now. And I feel very, very bad for Tampa Bay. I feel bad for all the fish in Tampa Bay right now, because of what is likely to happen over the next few weeks. Does anybody have any questions about that story, or how the microbes play in? You don't think about phosphate and nitrate as necessarily being toxic. You hear the word arsenic, or you hear the word mercury, and you or lead, and you think, oh yeah, that's a toxin. But nitrate and phosphate are toxic in the sense that they are toxic to the environment, because those are molecules that feed microbes. Microbes are going to metabolize those two molecules and use them to make ATP, and use them to grow, and use them to divide. And the normal balance that occurs in nature is going to be disrupted. And you're going to get these blooms, they call them, blooms of microbes growing. Those microbes tend to grow. And then they all tend to die. Once all the nitrate and the phosphate is used up, all the microbes die, and it sets up a toxic condition to all the other organisms that live in that body of water. It's very, very sad. All right, so a story in the news directly related to microbiology. I am going to just remind everybody of our schedule this week. I'm just going to pull up our Blackboard page right here. If you didn't see the announcement I sent over the weekend, be sure to take a look at that. I decided to extend an offer to students who may have some work that they have not submitted. There are certain things that I've decided to accept, even though the 1 week late period is already over. So if you have not seen this announcement, please read through it just in case it applies to you. I'm going to go now and scroll down to this week, which is the week of April 5th. So just a reminder that you have a lecture exam today that you need to get submitted by 8 p.m. This exam covers the 2 microbial metabolism lectures and the 2 microbial genetics lectures. We, as I said, on Wednesday, we're going to talk about your laboratory assignment called Unknown Microbe Identification. So there will be no lecture topics for you to view this week. This will be a lab heavy week. We'll be doing respiration today. We'll talk about the unknown assignment on Wednesday, and then you'll have lab questions due Sunday night as usual. If you scroll down in this content folder for this week, Of course, here's the lecture exam. Here are the slides for the lab. This is a folder that contains all of the documents that you'll need for the unknown microbe assignment. But again, we're going to talk about that on Wednesday, so don't worry about that right now. And here is the lab homework for this week. I am working on your pathogen assignment. I'm grading those. I anticipate those will be all graded by the end of the week. Remember, you're going to get those back, that first draft back, and I'm going to give you comments on your first draft to help you revise it and make a final draft. And then you'll be submitting that final draft to me in a couple of weeks. Part of the assignment, in other words, for this pathogen project is to give you an opportunity not only to practice your writing, but also to revise your writing according to editorial comments. So again, those will likely be returned to you by either the end of this week or the beginning of next week. All right. Any questions? Any questions about schedule? Any questions about anything? If not, what I'm going to do is open up our laboratory slides for today, and we'll talk about respiration. So, over the past couple of weeks, we've talked about carbohydrate catabolism. We've talked about fermentation. We've talked about protein catabolism. A lot of metabolic testing that we've been doing in order to discover what kind of metabolic abilities different microbes have, because we can use those results to help identify microbes. Today, we're going to be talking again about cellular respiration, and we'll be focused in a little bit on anaerobic respiration, a test that is specifically geared towards anaerobic respiration. Remember, when we use the term oxidative to describe a catabolic pathway, a pathway where a molecule is being broken down, that word oxidative does not have anything to do with oxygen. It refers to oxidation reduction reactions. And remember, an oxidation reaction involves the loss of an electron from an atom or a molecule, and reduction involves the gain. And during pathways like cellular respiration, it's a long, long series of oxidation reduction reactions. And of course, at the bottom of the electron transport chain, there's going to be a final reduction that occurs. A final molecule called the final electron acceptor is going to get reduced. That happens at the bottom of the electron transport chain. We've talked about how diverse microbes are. We've said many of them can do cellular respiration and oxidative phosphorylation to make ATP. We've talked about how they also do substrate level phosphorylation to make ATP. We've said they can do cellular respiration aerobically, some of them. Some of them can do it anaerobically. We've said some microbes can ferment. And of course, during fermentation, they use substrate level phosphorylation to make ATP. And of course, some microbes can do a mix of these pathways. Some microbes are capable of all kinds of mixes and matches of pathways, depending on the environment they find themselves in. We know that things like the OF test help us know if a microbe can do oxidative catabolism aerobically. Remember, we looked at an oil-covered tube and an open air tube. And in that open air tube, we watched for a color change, which would indicate that the microbe had done aerobic cellular respiration. The oxygen that microbe is using to do aerobic respiration, that oxygen is the final electron acceptor. And it's going to get reduced when it gains those electrons that came out of glucose. It's going to combine with those electrons and it's going to make water. It's also going to combine with a little hydrogen. I didn't put that in here, but I will. A little bit of hydrogen as well. So if you think about that long pathway of reactions that we call aerobic cellular respiration, it starts off with glucose getting oxidized and it ends with oxygen getting reduced. All of the electrons that are in glucose get released as glucose gets broken down. And all of those electrons are going to be fed into the electron transport chain so that the energy that they carry can be harvested. At the end of the electron transport chain, after all the energy has been harvested from them, they have to go somewhere. Because glucose got oxidized, something has to get reduced. And at the end of all of the reactions, it's going to be oxygen that gets reduced. Now, there's a part of this pathway that we haven't really talked about yet, and that's part of what's happening in the electron transport chain. The electron transport chain, if you recall from lecture, is this structure that we find in the membrane of the cell that's made out of a bunch of different proteins. And the electrons are quite literally being passed from one protein to the next protein to the next protein through this so-called electron transport chain. Now, there's a particular kind of molecule called a cytochrome that helps pass the electrons from protein complex to protein complex. Here's that term right here, cytochrome. So in order to get the electrons down the electron transport chain, we need a helper molecule to pass them along from protein to protein to protein. And that's what cytochromes do. Now, it turns out there are 4 kinds of bacterial cytochromes. There are 4 classes of bacterial cytochromes, and it depends on what species you're looking at as to what kind of cytochrome class they use in their electron transport chain. There's what are called cytochrome A, cytochrome B, cytochrome C, and cytochrome D. And again, this is a trait. This is a trait that different species of bacteria have, the particular type of cytochrome that you're going to find in their electron transport chain, assuming that they're able to do cellular respiration. So I have cytochrome C highlighted here because we're going to do a test today that tests for the presence of this particular type of cytochrome in a microbe, Cytochrome C. Now, when it comes to anaerobic organisms, remember, anaerobes that do cellular respiration, they're doing the exact same series of reactions. Glycolysis, followed by pyruvate processing, followed by Krebs cycle. All of the electrons are going to be sent down the electron transport chain to some final electron acceptor. The difference is that it's not oxygen in an anaerobe. It's going to be some other molecule. It's going to be an inorganic molecule, a molecule that does not contain carbon, but that does contain oxygen. So things like nitrate, sulfate, carbonate, not oxygen gas. That's what the aerobes use, but some other oxygen-rich inorganic molecule. Most anaerobes use these. I do want to just mention, so you're familiar, that there are some microbes, some anaerobes, who can also use other types of final electron acceptor molecules. Some microbes use organic final electron acceptors, in other words, carbon-containing Examples of those would be things like fumarate, triethylamine. There's a molecule called DMSO. When you read source materials, textbooks, and so on, it is possible for a microbe to do anaerobic cellular respiration and use one of these organic molecules as its final electron acceptor. But most anaerobes are using nitrate, sulfate, or carbonate. And among these, the most common is nitrate. So nitrate is a very, very common final electron acceptor molecule in the electron transport chain. Now, I showed you a minute ago that when oxygen accepts electrons at the bottom of the electron transport chain, that's the E minus, and also some hydrogen ions, water gets formed. That's what we call the pathway of reduction for oxygen gas. Now I want to show you what the pathway of reduction looks like for nitrate. So this nitrate molecule is going to accept electrons, and it's going to produce a molecule called nitrite. So nitrate and nitrite. It's also going to produce some water. So nitrate can be reduced to nitrite. That's one pathway that these anaerobic organisms use at the end of their electron transport chain. But there is a little complication here. There's a little bit more to the story for organisms that use nitrate. Some of them can take the nitrite molecule and they can reduce it even further. In other words, nitrite can also accept additional electrons and become a molecule called nitrous oxide. And it doesn't even end there, because nitrous oxide can also be reduced. It can accept electrons and create a molecule called nitrogen gas. So some microbes, their pathway of reduction for nitrate ends at nitrite. Nitrate gets reduced to nitrite. Other species can then take the nitrite and reduce it to nitrous oxide. And still other species can take the nitrous oxide and reduce it to nitrogen gas. Once again, we see a tremendous amount of diversity in microbes. Aerobic organisms that use oxygen gas as their final electron acceptor, they can only reduce it to water. They can reduce the oxygen gas into water. But anaerobic bacteria that are able to use nitrate as their final electron acceptor, they can do one of three different pathways to reduce the nitrate. Either nitrate to nitrite, nitrite to nitrous oxide, and even nitrous oxide to nitrogen gas. Three different ways. So we have to have a way to test. We have to have a way to test microbes so we can see which pathway they're using, if any, of those anaerobic pathways. Now, I also put on here the pathway for sulfate. That's what this molecule is. And for carbonate. You can see these pathways are a lot simpler. When sulfate gets reduced, when it accepts electrons, it also takes on, again, some hydrogen ion. You get hydrogen sulfide and water. And when carbonate gets reduced, and it takes on electrons and hydrogen ion, you get methane and water. So you may have heard that there are microbes that can release hydrogen sulfide. You may have heard that there are microbes that can release methane. Those are anaerobic organisms that are using either sulfate or carbonate as their final electron acceptor molecules. But nitrate is the most common. Now, I just put a little note down here for those of you who might be taking chemistry right now. These equations aren't balanced, but it's not what's important to us in this course. It's more important that we know what's going in and what's coming out. So this today for this exercise, we're going to be looking at a couple of different tests. We're going to first look at a test called the Oxidase test. This is the test that's going to look for the presence of an enzyme in the electron transport chain called Cytochrome C Oxidase. This is an enzyme that is going to help in this process of cytochrome C transferring electrons from protein to protein to protein in the electron transport chain. This enzyme is going to actually catalyze the oxidation of cytochrome C. In other words, cytochrome C in the electron transport chain is going to be located near one of these protein complexes. And as the electrons come into that protein complex, they will then be handed over to cytochrome C. In other words, cytochrome C is going to get reduced because it's going to gain electrons. Now, the cytochrome C has to give them to the next protein complex, the next step, if you will, in the electron transport chain. In order for it to do that, remember, if it's going to lose electrons, if it's going to give them away, it has to get oxidized. And it takes an enzyme to do this. And that enzyme is called cytochrome C oxidase. So this test, the oxidase test, is looking to see if a microbe has cytochrome C oxidase in it. Now, this is another one of those tests, like the catalase test, that's very quick and easy to perform. All you need to do is grow up some colonies on an agar plate. You're going to use a sterile cotton swab or a plastic loop to pick up some of those colonies, to pick up some of that bacterial cellular material. And then you're going to expose that material, those cells, to what's called the oxidase reagent. So you're going to drip some of this reagent onto that colony material, and you're going to look for a color change. If cytochrome C oxidase is present, you're going to get a color change to blue. It's actually more of a purple blue. I'll let you make up your mind what color it is. But it's a chemical called indophenol blue. So you're going to see, again, a purpley blue color change if cytochrome C oxidase is present. Cytochrome C oxidase is found in cells that use oxygen as their final electron acceptor. Remember, there are 4 classes. There's A, cytochrome B, cytochrome C, and cytochrome D. Microbes that use cytochrome C and have cytochrome C oxidase available, they're using oxygen as their final electron acceptor. Now, the other test that we're going to be doing today is called the nitrate reduction test. Now, this test is quite complex. This test has multiple steps to it, so we have to know how to evaluate the results after each step. I'm going to come back to this test in just a moment, but first, I want to show you what an oxidase test looks like. So remember, you're going to grow up the microbe. You can see that there's something growing on this quadrant streak plate, this agar plate. Most of the time we use a sterile cotton swab to just scoop up a little bit of this material. We're going to scoop up some of these colonies off of this plate. We want to get some cells onto the cotton swab. Then you're going to take the oxidase reagent. It comes in a little bottle, and you're going to drip it down onto that colony material on the swab. You're going to get an immediate result. If you see no color change, like here on swab B, that's a negative reaction for oxidase. If you get this purpley color, and again, some people see this as a blue, but if you get this blue purpley color, that's a positive reaction for Cytochrome C Oxidase. Now, here's an interesting thing about this test from a practical standpoint. The reagent that you're going to drip onto the cells on your swab, the reagent doesn't last very long. Usually it gets sold in these little bottles because once you open the bottle to start using it, in other words, once you break the seal, the oxidase reagent is only going to last for a relatively short amount of time, like a week or two. So even if you put the cap back on when you're done with it and you handle it very carefully, it's going to start breaking down. It's going to start going bad very quickly. So if you're in a laboratory that doesn't do a lot of oxidase tests, it's hard because you have to throw the reagent away after like 10 days or 2 weeks because it goes bad. So that led to the development of something called oxidase test strips. And these are actually quite common in microbiology laboratories. So instead of having a little squeeze bottle with the reagent in it, instead, you can get these little test strips that have the reagent already in the test strip. So all you have to do is scoop up some colonies from the agar plate and smear them onto the test strip and look for a color change. And that's what you're seeing in this image right here. So these are what the test strips look like. It's just a piece of paper that's been soaked in the oxidase reagent. And you take some of the colony material off of the plate and you just smear it onto the test strip like this. You can see this person has done it on both ends. And if it turns purple like this, and it will happen quickly, if it turns purple, you've got a positive test. If it doesn't, you've got a negative test. I will also mention it is possible to take some oxidase reagent in the bottle, the liquid reagent, and just drip it right onto the plate. That's what this person has done. In other words, you don't have to scoop up the cells onto a swab. You could just drip the reagent down onto the colonies and look for the purple color change. Most people don't do that because most people, they grew up this plate, so they're going to use those organisms for a bunch of different tests. So you wouldn't want to like ruin your plate by doing your oxidase test directly on the plate. So most people are going to scoop up cells onto a swab and drip the liquid reagent onto the swab, or they're going to scoop up cells and smear it onto the test strip. The beauty of the test strip is that those last longer. When you buy the liquid oxidase reagent, it goes bad really fast. So if you're a lab doing a lot of these tests, that's probably not a problem for you. But if you're a lab that doesn't do them very often, you probably don't want to buy the liquid. You'd want to buy the test strips instead. It's quick. It's a quick test, just like the catalase test with the bubble formation. But it gives us some important information about the microbe. All right, let's switch gears now and talk about this nitrogen reduction test. All right, now, like I said a minute ago, this is a complicated test. So be sure that you jot down any notes that you need so that you can understand this test later on when you go back to study. This test is going to test for the ability of a microbe to reduce nitrate to nitrite. If an organism can reduce nitrate to nitrite, we're going to get a positive test. Now, this test is based on a very basic culture medium, nutrient broth. But we're going to add some nitrate into it because we want to see if the microbe can reduce this nitrate. It's a form of nitrate called potassium nitrate. That's not really important, but we're putting nitrate in there because we want to see if it can get reduced by the microbe. So you're going to inoculate this broth and incubate it for an unusually long amount of time, really, 48 hours. 48 hours. We normally incubate things for 24 hours. There's a reason that we do this. It takes a while. It takes a while for some of these metabolic processes to occur to the point when where they are measurable. All right, we're going to look for a color change here like we do with a lot of tests. And in order for the color change to be evident, to be prominent and easy to see, you've got to give the microbe a full 48 hours to grow and metabolize. So it's an unusual one because it's 48 hours that we're going to incubate. Now, when I take the tube out of the incubator, after 48 hours, I'm going to add 2 different reagents to the tube. This, again, I told you this test is kind of complex. These reagents, they come in separate little squeeze bottles. Most people refer to them as reagent A and reagent B. We really don't need to know what the chemicals are. You just need to know that we're going to add 2 different chemicals into that tube. And we're going to look for a color change. If the organism that's growing in that tube has reduced the nitrate to nitrite, the tube is going to turn red. That's the first thing to know. That is a positive nitrate reduction test. Now, if I add reagent A and reagent B and I see no red color in the tube, I'm not done yet. And this is where it gets complicated. I'm going to add a 3rd chemical into the tube, and it's zinc. It's the zinc metal in a particular chemical form. Now, what is zinc going to do? Well, zinc as a metal is also able to reduce nitrate into nitrite. Now, this is where it gets complicated, so pay attention closely. If I put zinc into that colorless tube and the tube turns red, that is a negative result. And here's why. I put reagent A in, I put reagent B in. If the microbe turned the nitrate into nitrite, that means those reagents are going to make the tube turn red. If I put the reagents in and it doesn't turn red, that means there's no nitrite in that tube right now. And that means that the microbe did not reduce the nitrate. If I add zinc in, the zinc will reduce the nitrate and turn it into nitrite, and the tube will turn red. So if I add zinc and the tube turns red, that's a negative result. The microbe didn't reduce the nitrate, the zinc did. I have just proven by adding zinc in and creating a red color that there was nitrate in that tube, and the zinc has now reduced it to nitrite. So the microbe wasn't able to do that reaction, but the zinc did it. So even though it turns red, it's because I added the zinc. The zinc reduced the nitrate, the microbe did not. That's a negative test reaction. This is the first time we've seen the same color mean 2 different things, right? If I add reagent A and reagent B and it turns red, That's a positive result for nitrate reduction. If I add zinc and the tube turns red, that's a negative result for nitrate reduction. It's confusing, I know. But that's how you interpret that test. When we add reagent A and reagent B in there, if that microbe was able to reduce nitrate into nitrite, in other words, if there's nitrate in there, reagent A and reagent B will turn it red. If I add reagent A and reagent B and I don't get red, I don't really know what's gone on in there. All I know is there's no nitrate in there. So I add the zinc. The zinc will take any nitrate and turn it into nitrite, and then I'll get a red color. That's a negative result because the zinc did it. The microbe did not do it. But wait, there's more to this test. Remember, we said that it is also possible for some microbes to reduce nitrate to nitrite and then reduce the nitrite. Some of them can reduce nitrite into nitrous oxide, and some of them can reduce nitrite to nitrous oxide and then to nitrogen gas. How do I know if that's happened? Well, if I put reagent A and reagent B in and I get no color change, that tells me there's no nitrite in the tube. If I then add zinc and I still get no color change, now I'm into one of these. Either the bacteria has taken the nitrite all the way to nitrous oxide, or it has reduced it all the way to nitrogen gas. How do I know? There's going to be a Durham tube in there. There's a Durham tube inside that test tube. So all I have to do is look for a gas bubble. If there's a gas bubble, it has gone to nitrogen gas. If there's no gas bubble, it has gone to nitrous oxide. Here's a summary. Take the tube out of the incubator, the nitrate reduction test tube, take it out of the incubator, add reagents A and B. The microbe may have taken the nitrate and reduced it to nitrite. If there's nitrite in that tube and I add reagents A and B, I'm going to get a red color. That's a positive test result. In other words, the reagents showed that there was nitrite present in the tube by turning red. If I add A and B and there's no red color, well, now I have to add the zinc, right? But there are really two explanations for this. If there's no red color after A and B is added, either there's still nitrate in the tube because the microbe couldn't use it, couldn't reduce it, or there's nitrous oxide in there or nitrogen gas in there. So I have to be able to distinguish which one of these is happening. I'm going to add the zinc because if nitrate was in the tube, the zinc is going to convert it to nitrite, and I'm going to get a red color, negative result. If I add the zinc and the tube stays colorless, now I'm going to look for the gas bubble because either that microbe has made nitrous oxide or it's made nitrogen gas. Remember the zinc, if you put zinc in that tube and it turns red, that's a negative because the microbe didn't reduce the nitrate, the zinc did. If I add zinc and there's no color change, then the nitrate was reduced to nitrite, but then it was further reduced, either to nitrous oxide or to nitrogen gas. All right, so If I put the zinc in the tube and it stays colorless, that's a positive test, isn't it? That microbe had to have reduced the nitrite. That's a positive test. It took nitrate and it made nitrite. It just didn't stop there. It then also went on to nitrous oxide and sometimes even to nitrogen gas. But that's a positive test, because in order to get either nitrous oxide or nitrogen gas, the first thing that had to have happened was the nitrate became nitrite. So red after A and B, that's positive. Red after zinc, that's negative. Colorless after zinc, colorless after A and B, and colorless after zinc, that's positive. I just have to figure out whether or not it went to nitrous oxide or nitrogen gas. And I do that by looking in the Durham tube. If there's a bubble, that microbe made nitrogen gas. If there's no bubble, it made nitrous oxide. Very complicated test. The most complicated test that we've talked about so far in terms of how you interpret it. All right. So let's do these tests. Let's look at some organisms and run these tests on them, this oxidase test and this nitrate reduction test. Some of these we've used before. Staphylococcus epidermidis we've used before. E. coli we've used before. Pseudomonas aeruginosa we've used before. Here's a new one for us. Streptococcus pneumoniae. We looked at an organism called Klebsiella pneumoniae. That was a gram-negative organism. Streptococcus pneumoniae is a gram-positive organism. It's a gram-positive coccus. So we have two organisms that are gram-positive and are cocci, and we have two organisms that are gram-negative, and they are rods. This is that slide that you saw before, how to interpret your oxidase test results. We're looking for that color change when the cells are exposed to the oxidase reagent. So, Streptococcus pneumoniae, Staphylococcus epidermidis, our two gram-positive cocci, they're going to be negative on the oxidase test. E. coli and Pseudomonas, those are our two gram-negative organisms. E. coli is also going to show a gram-negative oxidase test. But Pseudomonas will show a positive test. Remember, if you're positive, if you're an organism that's positive on the oxidase test, what that means is your cells have this enzyme in them called Cytochrome C Oxidase. And that enzyme helps Cytochrome C get oxidized, lose its electrons, or donate its electrons back into the electron transport chain. It tells us that Pseudomonas uses oxygen at the end of its electron transport chain. That also is what we know from that positive result. So Pseudomonas must be able to do aerobic cellular respiration. What's important to know, though, is that that doesn't mean it's the only pathway that Pseudomonas can do. When you get a test result that says this organism is able to do this one pathway, that's great. You know something new about that organism. But you have to be careful not to think that that's the only pathway that that organism can do. Pseudomonas aeruginosa might also be able to do anaerobic cellular respiration. It might also be able to ferment. This test is only for that one pathway. So you have to be careful. You have to remember that when you interpret that result. Okay. The other three organisms, the Streptococcus Pneumoniae, the Staphylococcus epidermidis, the E. coli, what do we know about them? They're oxidase negative. Remember what it means. What they don't have is Cytochrome C Oxidase. You cannot say that therefore they must not do aerobic cellular respiration. You can't say that because they might be doing it with a different type of cytochrome. They're not using Cytochrome C, but they might be using A or B or D. We don't know. All we know is what they're not using. They're not using Cytochrome C. Okay? So again, it's really important when we see test results that we know how to interpret them. All right. Here's our nitrate reduction test. I wanted to show you this image over here on the right. This is what I mean when I say that red color. It's a very obvious red color. This medium starts off as a very boring beige. It's just a nutrient broth that we add a whole bunch of nitrate to. So you're just gonna inoculate this broth, this liquid. You're gonna incubate it for 48 hours. You're gonna add reagents A and B when you take it out of the incubator, and you're looking for this. You're looking for this red color. After I add A and B, if it turns red, I've got a positive test for nitrate reduction to nitrite. In other words, that red color, after I add A and B, is telling me that there is nitrite in the tube. And the only way nitrite got in there is because the cells made it. They took the nitrate that we put in this tube and they reduced it and created nitrite. And reagents A and B are showing me the nitrite by turning red. If I put A and B in there and the tube stays beige or colorless, as we usually call it, I got to do more work, don't I? So that's what this is about. This is a little flow chart that comes out of a textbook that helps us interpret this test. Now, they don't call the reagents A and B. They've actually named the reagents. You do not need to know the name of these reagents. Feel free to call them A and B. You take the broth tube out of the incubator after 48 hours, you add A and B into it. If it turns red, you've got a nitrate reduction test, positive result. If it stays colorless, well, now you got to do something else. Take that colorless tube and add zinc in. If it turns red after the zinc goes in, that's a negative result because the microbe didn't reduce the nitrate, the zinc did. If it remains colorless after you add the zinc, now you've got a positive test, but you have to figure out what the final product was. So you're going to look into the Durham tube. You can't really see it in this picture, but there is a Durham tube upside down in both of these tubes. Look for the gas bubble. Look for the gas bubble. If there's a gas bubble in that tube, that colorless tube, you know that the organism reduced nitrate to nitrite, then to nitrous oxide, then to nitrogen gas. If there's no gas bubble in this colorless tube, you know that the organism reduced the nitrate to nitrite and then to nitrous oxide. Positive test either way, but two different products that are in there, potentially in there. All right, let's look at our results. So this is a busy looking picture, I know, because of the names being typed onto the tubes, but this tube is the Streptococcus Pneumoniae. Here's the Staphylococcus epidermidis. Here's the E. coli. Here's the Pseudomonas. And you'll notice that there's some text added in here because, of course, we haven't seen what's been done to these tubes. Streptococcus pneumoniae tube turned red after the zinc was added. Staphylococcus epidermidis and E. coli, they turned red after reagent 1 and reagent 2 were added, or A and B, as we usually call them. Pseudomonas, it stayed colorless even after the zinc was added. And we have to check out this Durham tube at least, and try to figure out what's in there. So what does it mean? Actually, I'm going to skip that slide for just a sec. We took these tubes out of the incubator. We added reagent 1, or A, and reagent 2, or B. 2 of the tubes turned red: Staphylococcus epidermidis and E. coli. These are positive results for the reduction of nitrate to nitrite. These cells growing in these tubes took the nitrate and reduced it and made nitrite. And reagent A and B turned red because there's nitrite in here. Okay, these 2 tubes stayed colorless. The Streptococcus tube and the Pseudomonas tube stayed colorless after A and B were added. So in both of those tubes, we added zinc. The Streptococcus pneumoniae tube turned red after the zinc was added. That is a negative result for nitrate reduction. The red color occurred because the zinc reduced the nitrate that was in here to nitrite. The microbe didn't do it. The microbe couldn't do it. So the zinc did it. And that's why we got the red color after zinc was added. Pseudomonas stayed colorless after zinc was added. Pseudomonas is also a positive test. The reason it's colorless is because there's no nitrite in here anymore. The nitrite was further reduced. Now, what was it reduced to? Was it reduced to nitrous oxide, or was it reduced to nitrogen gas? I know it's hard to see in here, but here's the Durham tube right here. And you can see a little line of liquid right there in white. There's gas at the top of this Durham tube. So Pseudomonas aeruginosa was able to reduce nitrate all the way to nitrogen gas. Positive test, right here. Positive test, negative test. There is one more thing to note, and I almost hate to mention this because it just adds confusion for some students, but if you think about it, when I took this Pseudomonas tube out of the incubator, before I added any reagents in, before I added any of the reagents, there was a gas bubble at the top of that Durham tube, wasn't there? So it is possible when you pull these tubes out of the incubator after 48 hours, it is possible to peek at the Durham tubes before you do anything else. If there's gas in the Durham tube, you know you have a positive result. And that result produced nitrogen gas. Why? Well, because that's the only pathway that would produce gas. I know what's happened in that tube. Gas has been produced, and the only gas that can get produced is from nitrate all the way down to nitrogen gas. So yeah, it is possible when you pull these tubes out of the incubator to already know you're positive before you put any reagents in. It's always a good idea then to make sure you're right. Put the reagents in, put A and B in. It should not turn red. Put zinc in. It should still not turn red. That will confirm what you think, that you've got a positive result. Always a good idea to confirm everything you think you've got, just to double check. Oh, good question. Good question about the color in the tubes. I forgot to mention that. So one of your sharp-eyed classmates has noticed that the red color is different, isn't it? The red color is different between these tubes and this tube. So remember, we looked at this slide and we said this is the red color that we expect to see. It's a very cherry red, if you will. It's a deep red color. It looks like this, doesn't it? And these are a different red, right? You have to think about what's in the tube. The reason you get the difference in red is because of what's in the tube in terms of reagents. So these two tubes, they turned red after reagent A and B were put in. You're going to get more of a orangey red sometimes if you get a positive result after the first reagents are put in. This tube also had reagent A and B in it, but it has zinc as well. When we put A and B in, it didn't change color. So we put zinc in and then we got the color change. So this is a long path for me to say the red color can vary depending on what combination of reagents are in the tube. The key point, though, is that you're looking for that color change. In other words, from colorless to a red color. Good question. And yes, it would be a really good idea to put a control tube in there too, right? To take a nitrate reduction broth tube, don't put any cells in it, and keep it with all of your test tubes. Put it in the incubator for 48 hours, and then use it as a comparison so that you can see that big color change. Good question. All right, we're out of time. Does anybody have any questions? I think it's very helpful to put a little flow chart for yourself in your notes, a little flow chart for the nitrate reduction test. If this happens, then do this. If this color results, then you have positive or negative. If not, do this. Just really spell it out for yourself. All right. All right, everybody, good job. Remember, you have a lecture exam to do today. No lectures to watch this week. You have lab questions to do this week, and you're now able to do those lab questions if you have time. Those lab questions are only about this lecture or this lab. So if you want to, you can complete those right now. They're not due till Sunday, but you have everything you need to answer them. On Wednesday, we're going to talk about the unknown microbe identification assignment. Okay? All right, everybody. I will see you next time. Have a good day.
Hey, Professor.
Yes.
Can I talk to you after class?
Sure. All right, everybody. We'll see you next time. Hey, what can I do for you?
I don't know if you saw my email. I sent you an email. I lost my dad, so that's why I haven't been in class.
Yes, I actually replied to you just this morning about that. I'm so sorry to hear that news. It's terrible news.
Yeah.
I hope you're doing all right.
Yeah, I'm doing all right. It's just so I was in Monday's class, but You remember Monday's class ended a little early because of the wind.
That's right.
Yeah, but I know I miss Wednesday
Uh-huh.
and Fridays, and we
We
had
didn't
assignments.
actually meet on Friday, so it was
Oh
really
yeah,
only Wednesday. Yeah.
yeah. So we did have assignments last week. I'm gonna pass them in this week with
Okay.
this week's work. I just wanted to like let you know that.
I appreciate that. Yeah,
And
that sounds good.
how many points will I lose off of that?
You won't, because of course you had a reason for why you weren't there. You weren't in
Okay.
class, and you have a reason for why the work is late. So don't worry about that. I won't take any points off of anything.
Okay.
Okay?
Yeah.
And again, I'm really sorry that you've gone through that. So challenging.
Yeah.
But I'm
Thank you
glad
so
you're
much.
back.
Yeah. Thank you.
All right, we'll see you next time.
Okay. Bye.
Bye-bye.